Welcome to this session of our science talk program. Our guest this week is Professor Michel Steinert. His specialty is infection biology. He is from the Institute of Microbiology at the Technical University of Braunschweig, and simultaneously he is also affiliated with the Turkish-German University. The subject of the talk today is very interesting. It is regarding it's an interdisciplinary approach to sustainable advance and safeguards for the health of humans, animals, and the environment. And essentially, the One Health approach that is going to be described by Professor Steinert emphasizes that the health of people in general is interlinked with our ecosystem and the species inhabiting in the ecosystem. In particular, the One Health approach was quite relevant given the recent global spread of H5N1 influenza and in particular the COVID-19 pandemic that we just lived. So the rise of antibiotic resistance and increasing frequency and the magnitude of harmful algae is also very important, in the applications of the One Health approach. In essence, the One Health approach brings together all aspects of public health, including the collaboration among professionals in various domains. That includes the medicine, the veterinary science, environmental science, and it also requires effective communications and coordinated policies in between these various disciplines. Professor Orkide Coşkuner, she is from Turkish-German University, and she is a very close associate of Professor Steinert. I invite her to give a brief introduction of the background of Professor Steinert. The floor is yours, Professor Coşkuner.
Thank you very much. So it is an honor and a great pleasure to introduce today Professor Dr. Michael Steinert from the Technical University of Braunschweig in Germany. He's currently the managing director of the Institute of Microbiology. Professor Steinert studied biology at the University of Würzburg and was a Max Buchner Research Stipend and a postdoctoral fellow at the Centers for Disease Control and Prevention in the United States of America. He then became a group leader at the University of Würzburg. He's a flying faculty member at the Turkish-German University and the Molecular Biotechnology Department. His scientific interests include human, animal, and plant health, and their shared environment from a complex relationship where the health of one species depends on others, which he calls One Health approach. Today, we are going to listen to Professor Dr. Michael Steinert's research on One Health approach. Welcome, dear Professor Dr. Michael Steinert. The stage is yours.
Okay, yeah, thank you very much. Thank you for the kind invitation. It's a great honor. And also for the kind introduction of my career. I will open the screen. I hope you can hear me and see the screen. So
we can see the screen, yes, thank you.
Perfect, thank you very much. Yeah, well, the topic of my talk is One Health in molecular infection biology. But before I start, and now I have to make sure I can skip this. I want to tell you where I'm from. So I'm from Braunschweig, and you see Braunschweig on this map. So it's basically in the north, and we are between the Heide and the Harz Mountains. And Braunschweig is a very old city, and currently we do have snow. But it's also a young city because we have many students. And we also have a kind of Turkish infrastructure in our town, which is highly which we enjoy a lot. Well, this is the building, the main building of our university, and now I'm sitting in this brick building, this red brick building, and this is from here where I start my presentation. So One Health, it is an integrated, unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystems. And we do have a number of challenges because our population is growing, we have increasing global mobility, we have intensive animal husbandry, climate change, loss of natural habitats and biodiversity loss. And this also has a strong influence on how host microbe interactions and especially on infections. And the threats we are facing is new emergent pathogens and a rapid spread of infectious diseases, zoonosis, and antibiotic resistance. And these global challenges need an answer. And this answer is teaching on one hand, but also a kind of engineering for health. And I want to address both topics, and we do this, for example, together with the Turkish-German University in Istanbul. And we do have a nice collaboration, and I really enjoy very much the interaction between the Turkish-German University and our university, and I enjoyed some highlights of the recent years very much. And it is not only working on the political level, we also do have flying students. And in the recent years, I had several guest researchers and students from the Turkish-German University in my laboratory, and I enjoyed this very, very much. And a lot of this was also organized by the Erasmus program, which is between our universities. So, but let's get more specific. Our answers to global health challenges. So I'm involved in 3 projects. The one is ash dieback. Another one is about honeybees, the influence on pesticides and pathogens and the honeybee fitness. And the third topic is Legionnaire's disease. The first 2 I will only address very quickly, and then we will go deeper into this human disease, which is Legionnaire's disease. So we are working on a biological control of ash dieback. Ash is a tree in Europe, and as you can see in this picture, these ash trees die because of a pathogenic fungus, Haemolycophysis fraxineus, which comes from Southeast Asia. And what we are doing in this project is that we are searching for fungal endophytes which produce secondary metabolites to protect the tree from this pathogen. You see how we isolate endophytes, we characterize the metabolites, we evaluate their inhibition potential, and then we analyze this biological control mechanisms on small ash trees, but also on bigger ash trees in greenhouses. Moreover, we are currently working on a vaccination of plants, which is quite a new principle. So we prime the plants with virulent strains, and we do see some resistance developing. The second topic I'm working on is honeybee fitness and the influence of pesticides and the influence of these treatments on pathogens and the health of the bees. As you may know, all animals do have a microbiome, and there is a kind of core microbiome. These are bacterial which live in the gut, for example, or in the beehive. And there's an accessory microbiome. These are microbes the animals collect from plants, for example. And they're quite important because they help to digest. They are providing essential nutrients. They are important for detoxification. They protect against pathogens and parasites. So basically, they are also important for immunity. And by applying pesticides, for example, they have quite a strong influence on the fitness of the honeybees. And we are interested in this. And as you see on this slide, you can see that certain fungicides, for example, which are applied on our fields, they move into the animals, they move into the hive, and then you find these compounds also in the larvae of, for example, of the bees. So there is a kind of transfer of these chemicals, and the situation is even more complex because depending on the sex, on the caste of the bee, males, females, the queen, the diet, environmental conditions, these pesticides do have an effect on the fitness of the bees. So it has an effect on the immunity, and the effects on immunity are also related to nutrition, for example. So it's a very complex network of influences: pesticides, antibiotics, pollution, climate change, and so on. And here we analyze, for example, the resistance of the microbiota against the plant protection compounds, but we also are interested in the stress level on the honeybees, in dysbiosis, which is a disturbance of the gut microbiome, for example. And we also analyze how much the bees are susceptible against certain pathogens, like, for example, Penibacillus larvae, which is the causative agent of American foul brood. And this is a disease which kills the bee larvae and makes the hive to collapse after some time of infection. So we are investigating if there are bacteria, for example, actinobacteria or phages, which can protect the honeybee from this American foul brood. And we also try to find inhibitory metabolites of microbes and plants to protect these bees. Well, let's move to my main topic, which is Legionnaire's disease. And I will highlight you the basics of this disease and the pathogen, which is Legionnaire pneumophila. And then I want to move on host pathogen interactions and then certain applications to stop this disease. So Legionnaire's disease was first documented in 1976 during a meeting of the American Legion Convention, and 182 cases of pneumonia occurred in this hotel in Philadelphia, and 19 patients died. And it took almost 1 year for the CDC to identify the pathogen. They found a gram-negative flagellated rod. It belongs to the gamma proteobacteria. And because Legionnaire's veterans were affected, they called this bacterium Legionella. And because they were suffering from a pneumonia, the name was Legionella pneumophila. Well, Legionella is a bacterium which is ubiquitous in water. It likes elevated temperatures and stagnant water. You can find them in biofilms and protozoa, and they are distributed by technical vectors like cooling towers, showers, air conditioning system, whirlpools, etc. And interestingly, Legionella in the environment parasitizes intracellularly within protozoa, which are, for example, amoeba living in aquatic habitats. These amoeba provide nutrients, they protect Legionella, they can use amoeba as a vehicle, and also this amoeba, when inhaled, can lead to human infection. And this is an overview. You see on the left-hand side a red-stained bacterium in the aquatic habitat, and you see green-stained this intracellular bacteria within a protozoa. And this is basically the infection cycle you find in the environment in water. The transmission then is by the technical vectors, for example, showers. And if we inhale this bacteria or amoeba filled with the bacteria can cause a severe infection. And then you almost see a mirrored picture. So you find extracellular Legionella in the extracellular matrix of the lung, but you also find intracellular multiplication in the alveolar macrophages, which are cells which should defend us from infection. But since Legionella learned how to survive within a protozoan, they can also survive within macrophages. So basically, the protozoa in the environment are kind of virulence school. And if we inhale the bacteria, the mechanisms which are successful in the protozoa are also successful in the macrophages in our lungs. What is the result of this infection? It is either Legionnaire's disease, which is a severe pneumonia, and we have about 20,000 cases per year in Germany. And the other disease is Pontiac fever, which is a self-limited non-pneumonic fever. And here the doctors are not able to isolate live bacteria from the patients. Well, now we try to understand Legionella infection. And this is not easy because you have a lot of extra and intracellular interactions which occur within the lung at the same time. You have different types of cells, you have acellular components of the lung. Moreover, you have a dynamic progression of the disease. So the beginning of the disease is different than the end of the disease. So it is a quite complex situation for a scientist. And to have a perfect model, we developed a human ex vivo tissue culture model. So here you see the sample preparation. So this is human lung tissue we get from hospitals. These are tumor patients. And then we are able to culture these tissue blocks for 72 hours in our laboratory. And for this time window, the tissue blocks are a kind of healthy and stable. And we can use these tissue blocks to mimic infection in the human lung. And we did a lot of histopathology of infected human lung tissue explants. And what we observed was that it really mimics very nicely the infection of a real patient. So we still see that macrophages are recruited. These macrophages take up Legionella within these blocks. And we also see how the lung tissue is destroyed by the pathogens. Yeah? So we, for example, see tissue disaggregation and the connective tissue in this lung tissue blocks is destroyed. And this gives us a nice possibility to compare, for example, reference strains of Legionella with new clinical isolates. We can also compare reference strains with genetic mutants we generate in our laboratory. We can use purified virulence factors, which we isolate in our laboratory and analyze the specific effect of one protein on the lung tissue. And one protein we analyze since a while is the zinc metalloprotease. It is the major secretory protein of Legionella, and it is known to be hemolytic and is also known to kill cells. So what did we do in our experiment? We started the culture of our human lung tissue explants, which you see in the upper row of our slide. And then we injected Legionella in the top surface of a tissue block. And then we analyzed how Legionella is transmigrating into the deeper layers of this tissue. And this we then analyze by microscopy. We do very thin slides of the tissues and analyze the layers. And what we find is that this protein, this Pro-A protein enhances bacterial transmigration into deeper tissue layers. So this protein helps the bacteria to spread in the lung tissue and to get deep into the tissue. We also did histological analysis of the Pro-A protein, and we found that it causes tissue damage. In these tissue pictures, you see on the left-hand side the control, and on the right-hand side, you see when we add Pro-A to the tissue. And you can see the difference after 24 hours. What we see is that the Pro-A protein causes severe tissue damage. But this is not the only thing. We also see that Pro-A increases the alveolar septal thickness. Again, on the left-hand side, the control, and on the right-hand side, we added 10 microgram Pro-A to the tissue, and you see how swollen the tissue is. So we were interested, why is the tissue so swollen, which is a sign of inflammation also. And we found that this protein is degrading collagen in the lung tissue. So Pro-A hydrolyzes type 4 collagen in lung tissue, and this destroys the basal lamina of the lung tissue. We were also interested to see what kind of effect does Pro-A have on the cells of the lung. And again, we used our human lung tissue explants. We cut them into blocks, and then we found, Pro-A promotes bacterial multiplication in the tissue. So it is not only responsible for transmigration in dispersal in the tissue, it also helps the bacteria to proliferate in the tissue. Another factor, and now we are getting more molecular, is the analysis of inflammation. So this bacteria, this Legionella, do have a flagellum, and flagella are shown here. It is a long filament, and it is responsible for the movement, for the motility of the bacteria. This flagellum consists on many Fla-A monomers. These are little proteins. And as long as the Fla-A proteins are a monomer and not aggregated in a filament, they cause inflammation. So it's a trigger of pro-inflammatory cytokines, and there's a pathway, is the toll-like receptor 5 pathway, and this pathway causes inflammation. Now the bacteria do an interesting trick, because they use the enzyme Pro-A to degrade monomeric Fla-A. So they degrade the trigger of this pathway. And this is very clever of the bacteria, because if they destroy monomeric Fla-A, then the immune system will not detect the bacteria easily. So they can camouflage themselves by destroying this trigger. And this is what Pro-A does. And so we could also show this effect not only in cells, but also in tissue. And by this, Pro-A reduces a Fla-A-dependent activation of this pathway, which otherwise would clear Legionella from the tissue. So it's a measure by which the bacteria trick the human immune system. Now, since we discovered that Pro-A is important for the transmigration of the bacteria in the tissue, the spread of the bacteria in the tissue, and that it is also important to confuse the human immune system, we now develop compounds which block this virulence factor of Legionella. So we found novel zinc binding inhibitors which can block the enzyme. And with this new compounds, we are able to protect the tissue. So the aim is we give antibiotics, and if these new compounds are in the clinic, we hope that we gain time, because we would then not only give antibiotics, but also a tissue protectant, a kind of pathoblocker. And this gives the doctors more time for antibiotic treatment on one hand, and it would also hopefully prevent lung damage, which can be a long-lasting problem for the patient. Now I move on to a second virulence factor, which is the macrophage infectivity potentiator of MIP. So this protein is important for the infection of macrophages, and in my introduction I mentioned already macrophages are the main target cells of Legionella. So this is the cells where the bacteria replicate, although they are the main or first line of defense of our immune system. And on the right-hand side, you see the histopathology of an infected guinea pig. And if we have a genetic mutant of Legionella, which is indicated by MIP negative, you see almost a healthy lung. Yeah, you see the alveoli, and it is good that you have this white space in between, because this is where the gas exchange in the lung takes place. This helps us to breathe. But if you have a wild type bacterium which carries the MIP protein, these alveoli are filled with liquid, with infiltrate. So basically our lung is full of fluid, and this makes the symptoms of pneumonia. And we could demonstrate that this MIP protein of Legionella is also responsible for the dissemination of the bacteria within the lung. And it also reduces the replication in the lung. So therefore, we were puzzled, what is the specific contribution of MIP in this lung tissue damage? So what we did is we isolated the MIP protein and added purified MIP protein to the lung tissue. And then we were very surprised, because we did not see any tissue damage. However, if we use a genetically engineered Legionella, which does not have MIP anymore, we do see a reduced tissue damage. And in the first moment, we thought this is a contradiction, but there may be a solution to this contradiction. Maybe the effect of MIP is more indirect. Maybe it just contributes to adhesion, or it helps other enzymes to do the damage. Now we are getting very molecular, and I will just briefly mention what we did. So we try to find out what are the interaction partners of MIP. And we did this by a co-immunoprecipitation, and then we found some proteins which interact with MIP, and we also find some compounds which could block this interaction. This was FK506. And so this was an interesting result, because now we know MIP is not damaging the tissue directly, but it uses other interaction partners and helps other interaction partners to damage the lung. Well, we got even more specific and analyzed certain amino acids and certain qualities of the protein. For example, this protein doesn't produce dimers and has many interaction partners which were not known before. And we also used modeling, is a computer-based analysis of interaction, and we could analyze dynamics of the protein with other proteins, and we could analyze how other proteins bind to MIP. But I don't want to go into too much detail, but I want to highlight the following. So we then again looked in the microscope and looked what does MIP do with the bacteria itself. And here you see transmission electron micrographs of Legionella pneumophila. And we found that if the MIP protein is knocked out genetically, or the MIP protein is blocked by a compound called FK506, then the bacteria again produce less flagella. And as we have learned from the ProA project, flagella is important for the immune system. So again, we see, okay, MIP is doing something with flagella. And we could observe that the MIP protein promotes flagellation of Legionella and then also of motility. And we could also see that this interaction can be blocked by FK506. Again, we use this molecular information to develop a compound which blocks the MIP protein. And again, two Turkish students of my lab, Zafer Karagöz and Can Ünal, were very successfully in analyzing new compounds which block this MIP protein and then eventually again can help to protect lung tissue during the treatment of Legionnaire's disease. And well, for us, it was very interesting to see that both virulence factors, ProA, which I discussed in the beginning of my talk, and MIP, they both target the flagellum and the host collagen, and they contribute to degradation of LAA. Both proteins promote bacterial transmigration and the damage of the tissue. And they both regulate virulence factors, including flagellation and the immune evasion. For us, it is an interesting starting point to, on one hand, further analyze, are ProA and MIP cooperative? Do they work coordinatedly? This we don't know yet. We also want to understand better how MIP is involved into the flagellation of the bacteria. But our main question for the future is, are ProA and MIP good targets for tissue protectant? So our main aim is to develop new compounds which block these virulence factors of Legionella. And then we want to achieve the protection of the infected lung tissue. And this will help the doctors to gain time for antibiotic treatment. So this is our future challenge and our future direction. And with this, I want to thank my cooperation partners from the hospital in Braunschweig, from our cooperation partners in Spain and from the Technische Universität Darmstadt, from the Julius Kühn Institute in Braunschweig, for the Helmholtz Center of Infection Research, and and my workers of my working group. And I thank you for your attention, and I will be happy to answer questions. Thank you very much. It was a great pleasure to be invited.
Thank you, Michael. Are there any questions, either from the YouTube channel or from the Zoom participants? Can
I
I
have a question.
Go ahead, Professor Orkide.
Yes. Can I ask a question?
Yes, sure.
Yes. We have been studying the role of intrinsic disorder proteins in viral infections and bacterial infections. And we found that many of the proteins have intrinsic disorder regions. Do these proteins have intrinsic disorder regions as well, or are they ordered structures?
They are ordered structures, as far as I know.
Mm-hmm.
But the interaction partners we don't know. So maybe in the interaction partners we do have this, and this would be really interesting to know.
Yes.
Yeah.
Yes. Thank you.
Yeah.
Professor Steinert, the One Health approach that you described, it requires coordination of many different stakeholders in the public health ecosystem. Could you please explain some of the difficulties you face maybe in Germany in implementing the One Health approach to the public health system within your country?
Yeah. Well, first of all, the One Health approach brings together experts from very different fields. So you have the veterinarians, you have the human doctors, you have scientists, you have people interested in ecology and biodiversity. And one major problem is already they do speak different languages, not in terms of English or German or Turkish, but they have a different understanding and they have a different level of looking at things. So one is looking more globally if you are interested in epidemiology, and we are looking deep into the molecules. Professor Weber is looking at atomic details and energy levels. And to bring this expertise together is really difficult because there is a lot of misunderstanding or not understanding. And I think this is one of the major challenge. And this was also a reason why I started to analyze this disease, for example, like Legionnaire's disease, on the tissue level, which is a macroscopic level. Then we went deeper into the cellular level. And from the cellular level, we went deeper into biochemistry and molecular biology. And yeah, in the recent years, we even went into computational biology, although this is not my personal expertise, but it does not work without. And to zoom in into more and more details and to be able to understand each level. This is what helps. But this is also the challenge.
What is the level of implementation of the One Health approach? I mean, what percentage of the objectives are accomplished, you think, in Germany? And what would your recommendations to Turkey would be to implement this in the Turkish public health ecosystem?
Well, I think, since the biodiversity is a global problem, the loss of biodiversity is a global level. It is very hard to give percentages because, it was for a good reason that I also mentioned teaching in the beginning. It's not only research, it's also teaching. If people understand the problem, this is, and that environmental protection is important. For example, we do see the Ebola outbreaks in Africa. Meanwhile, it is known that Ebola outbreaks are linked to the elimination of forest in Africa. It brings animals and humans closer together. Wild animals don't have enough space anymore, so there's more contact with wilderness, and then viruses can jump and spill over into the human population. So we have to really understand on all levels, and I would say all levels are important. It's very difficult to say this or this level is more important, but we do need education that people understand this. And this is also something I do as a kind of third mission, we call it. So I do talk to schools, for example, and to implement this idea in schools. It's also important to talk to politicians, because, well, there's always economic pressure, and then, well, the environment is often seen as a resource you can just use, but you don't have to protect. And so I think with good basic science, you have to go into schools, into politics, into all kinds of social in all kinds of social levels to get this idea across.
Sesiniz gelmiyor Orkun Hocam.
Sorry, I think I was muted. My wife is responsible for the public health implementation, also policy development in certain districts of Istanbul. Might be nice, if when you come here, to Istanbul, I would introduce you to her department and discuss maybe opportunities, for collaboration, in this regard.
Will be my pleasure.
Yes.
I will be in Istanbul in May.
Okay, looking forward to meeting you. Okay. So if there are, Orkide Hocam, are there any further questions?
Not from me. Thank you very much. It was a great talk.
Okay, so thank you for introducing Professor Steinert, Orkide Hocam, and looking forward to further collaboration in the near future, hopefully. And thank you for this nice presentation.
Thank you very much. Thank you, and goodbye.
Bye.